Humans have long held a fascination for the human brain. We chart it, we've described it, we've drawn it, we've mapped it. Now, just like the physical maps of our world that have been highly influenced by technology, think Google Maps, think GPS, the same thing is happening for brain mapping, true transformation. So let's take a look at the brain. Most people, when they first look at a fresh human brain, they say it doesn't look like what you're typically looking at when someone shows you a brain. Typically, what you're looking at is a fixed brain. It's gray. And this outer layer, this is the vasculature, which is incredible around the human brain. This is the blood vessels. 20% of the oxygen coming from your lungs, 20% of the blood pumped from your heart is servicing this one organ. That's basically, if you hold two fists together, it's just slightly larger than the two fists. Scientists, sort of in the end of the 20th century, learned that they could track blood flow to map non-invasively where activity was going on in the human brain. So, for example, they can see in the back part of the brain, which is just turning around there, there's the cerebellum. That's keeping you upright right now. It's keeping me standing. It's involved in coordinated movement. On the side here, this is the temporal cortex. This is the area where primary auditory processing, so you're hearing my words, you're sending it up into higher language processing centers. Towards the front of the brain is the place in which all of the sort of more complex thought, decision-making is the last to mature, sort of late adulthood. This is where all your decision-making processes are going on. It's the place where you're deciding right now you probably aren't going to order the steak for dinner. So if you take a deeper look at the brain, one of the things, if you look at it in cross-section, what you can see is that you can't really see a whole lot of structure there. But there's actually a lot of structure there. It's cells and it's wires all wired together. So about 100 years ago, some scientists invented a stain that would stain cells. And that's shown here. In the very light blue, you can see areas where neuronal cell bodies are being stained. And what you can see is it's very non-uniform. You see a lot more structure there. So the outer part of that brain is the neocortex. It's one sort of continuous processing unit, if you will. But you can also see things underneath there as well. And all of these blank areas are the areas in which the wires are running through. They're probably less cell dense. So there's about 86 billion neurons in our brain. And as you can see, they're very non-uniformly distributed. And how they're distributed really contributes to their underlying function. And of course, as I mentioned before, since we can now start to map brain function, we can start to tie these into the individual cells. So let's take a deeper look. Let's look at neurons. So as I mentioned, there are 86 billion neurons. There are also these smaller cells, as you'll see. These are support cells, astrocytes, glia. And the nerves themselves are the ones who are receiving input. They're storing it. They're processing it. Each neuron is connected via synapses to up to 10,000 other neurons in your brain. And each neuron itself is largely unique. The unique character of both individual neurons and neurons within a collection of the brain are driven by fundamental properties of their underlying biochemistry. These are proteins. They're proteins that are controlling things like ion channel movement. They're controlling who nervous system cells partner up with. And they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genomes. We each have 23 pairs of chromosomes. We get one from mom, one from dad, and on these chromosomes are roughly 25,000 genes. They're encoded in the DNA, and the nature of a given cell driving its underlying biochemistry is dictated by which of these 25,000 genes are turned on and at what level they're turned on. And so our project is seeking to look at this readout, understanding which of these 25,000 genes is turned on. So in order to undertake such a project, we obviously need brains. So we sent our lab technician out. We were seeking normal human brains. What we actually start with is a medical examiner's office. This is a place where the dead are brought in. We are seeking normal human brains. There's a lot of criteria by which we're selecting these brains. We want to make sure that we have normal human between the ages of 20 to 60. They died a somewhat natural death with no injury to the brain, no history of psychiatric disease, no drugs on board. We do a toxicology workup. And we're very careful about the brains that we do take. We're also selecting for brains in which we can get the tissue, we can get consent to take the tissue within 24 hours of time of death. Because what we're trying to measure, the RNA, which is the readout from our genes, is very labile, and so we have to move very quickly. One side note on the collection of brains, because of the way that we collect and because we're requiring consent, we actually have a lot more male brains than female brains. Males are much more likely to die accidental death in the prime of their life, and men are much more likely to have their significant other spouse give consent than the other way around. So the first thing that we do at the site of collection is we collect what's called an MR. This is magnetic resonance imaging, MRI. It's a standard template by which we're going to hang the rest of this data. So we collect this MR, and you can think of this as our satellite view for our map. The next thing we do is we collect what's called the diffusion tensor imaging. This maps the large cabling in the brain. And again, you can think of this as almost mapping our interstate highways, if you will. The brain is removed from the skull, and then it's sliced into 1 centimeter slices. And those are frozen solid, and they're shipped to Seattle. And in Seattle, we take these, this is a whole human hemisphere, and we put them into what's basically a glorified meat slicer. There's a blade here that's going to cut across the section of the tissue and transfer it to a microscope slide. We're going to then apply one of those stains to it, and we scan it, and then what we get is our first mapping. So this is where our experts come in and they make basic anatomic assignments. You could consider this state boundaries, if you will, those pretty broad outlines. From this, we're able to then fragment that brain into further pieces, which then we can put on a smaller cryostat. And this is just showing this here. This is frozen tissue, and it's being cut. This is 20 microns thin, so this is about a baby hair's width. And remember, it's frozen, and so you can see here old-fashioned technology of a paintbrush being applied. We'd take a microscope slide, and we very carefully melt onto this slide. This will then go into a robot that's going to apply one of those stains to it. Okay. And our anatomists are going to go in and take a deeper look at this. So again, this is what they can see under the microscope. You can see collections and configurations of large and small cells in clusters in various places. And from their expertise, they understand where to make these assignments, and they can make basically what's a reference atlas. This is a more detailed map. Our scientists then use this to go back to another piece of that tissue and do what's called laser scanning microdissection. So the technician takes the instructions, they scribe along a place there, and then the laser